During part 2 of the muscles unit, we are going to go over contraction and relaxation of skeletal muscle fibers. During the last part, we talked about the structure of the sarcomere. It is important to note that during a contraction and relaxation cycle, the length of the thick and thin filaments is not changing. However, the length of the sarcomere shortens during a contraction cycle. Now, during muscle contraction, the thick filaments essentially walks or slides across the thin filaments, or the thin filaments slide across the thick filaments. So the Z discs essentially move towards the M line. So the length of the filaments itself is not changing, but the filaments are sliding against each other. So let's see how that takes place. It occurs in 4 steps. In the first step, the excitation of the sarcomere takes place. And this happens when a neuron brings in the efferent signal to it. So a neuron is a efferent or effector neuron, brings the excitation signal to a muscle fiber. So when this signal comes to the muscle fiber, an electrical signal has to activate the regulatory proteins. You should recall the regulatory proteins that we talked about in the last part of this chapter. Those 2 regulatory proteins are troponin and tropomyosin. These allow the contraction cycle to take place. The next part is the actual sliding of the thick and thin filaments. So that is the contraction cycle. The last step is the relaxation. So how the neuron brings about the signal will be discussed in detail in chapter 12, which is our nervous tissue chapter. So basically, the action potential travels down the length of the neuron, and I will explain this based on images in just a little bit. But the plasma membrane is charged because there's more negative charge on the inside of the neuron, and there is more positive charge on the outside. So there is a temporary change. So the neuron has a tail, and that tail is called the axon. So here's what the neuron looks like. The inside of the neuron has a negative charge, and the outside of the neuron has a positive charge. So there's a temporary shift wherein the inside becomes positively charged and the outside becomes negatively charged. And this temporary shift travels down the tail or the axon, and towards the terminal end, there's a bulbous process. This bulbous process is called the synaptic end bulb. So let's see what this looks like. So where the synaptic end bulb fuses with the muscle fiber, that spot is called a motor unit. So skeletal muscle has to be stimulated by somatic motor neurons. Anytime a muscle is fused with a neuron, it is called a motor neuron. So that type of neuron that goes to a muscle is called a motor neuron. And anytime a neuron goes to a skeletal muscle, it is called a somatic motor neuron. So it's a point that you need to note. Anytime a neuron goes to a skeletal muscle, it is called a somatic motor neuron. Now, a motor unit is where the synaptic end bulb fuses with a muscle fiber. So this is what it looks like. So take a look at this motor unit right here. When a motor neuron and the muscle fiber innervates, it is called a motor unit. So here's your muscle fiber right here, and this is a neuromuscular junction. So you have, this is the spinal cord. From here, this green neuron is going right here, and it's going right here and right here. It's forming a junction. The neuron is forming a junction with the muscle fiber, hence the term neuromuscular junction. And it is a motor neuron because it is going to a muscle fiber. Now, motor units can be different size. You can have smaller motor units, which can involve 2 to 20 muscle fibers, and you can have larger motor units, which go to larger muscles, such as the calf muscle. The calf muscle is the gastrocnemius muscle that can have thousands of motor units. The smaller motor units can go to muscles such as your eye muscles that control the movement of the eyeball, the lateral rectus, the medial, the superior inferior rectus muscle. So they can be different sized motor units. Here's a scanning electron micrograph of a neuromuscular junction. So it's essentially a synapse. A synapse is where there's going to be some kind of action between action potentials is going on. So right here is the somatic motor neuron. You can see the neuromuscular junction is right here. You can see the synaptic end bulb. You can see the striations over here. Here's your muscle fiber. And right here is where the neuromuscular junctions that are happening. A synapse is where a neuron meets a target cell. This target cell can be either another neuron or it can be a muscle fiber. So this diagram is something that you guys really need to understand. I'm going to spend a lot of time explaining this diagram. So this is where excitation is going to take place. So let's understand what is going on in this diagram. Let's understand what is happening in step 1 of the muscle contraction. So this is the axon. This is part of the somatic motor neuron. So action potential is traveling. This arrow represents the direction of action potential. So the signal, the nerve signal is traveling down the length of the axon. When it reaches this bulbous portion, aka the synaptic end bulb, something happens. That something is that calcium channels open up. Where's my pointer here? Okay. So as the nervous signal, as the action potential reaches the synaptic end bulb, calcium channels are opened up. So these purple channels right here are the calcium channels. Calcium channels are opened up. So as calcium channels are opened up, calcium flows into the synaptic end bulb. So look at these purple beads right here. Those are calcium ions. So calcium channels open up, and because of that, calcium enters into the synaptic end bulb. So that's step number 1. Action potential reaches synaptic end bulb, calcium enters synaptic end bulb because calcium ion gates have opened. Step number 2, because calcium has now entered the synaptic end bulb, that triggers something. So inside the synaptic end bulb, sitting in little storage vesicles, is a neurotransmitter. This neurotransmitter is called acetylcholine, or ACH. It's represented as ACH, acetylcholine. So as soon as calcium enters the synaptic end bulb, it triggers the exocytosis of acetylcholine. So acetylcholine is just hanging out, doing its thing in little storage vesicles. And as calcium comes in, these storage vesicles are exocytosed out of the synaptic end bulb. Now, where exactly are they exocytosed? They're exocytosed into this little gap between the synaptic end bulb and the muscle fiber right here. So take a look at this. This blue space right here is called the synaptic cleft. The synaptic cleft is that little space between the junction between the neuron and the muscle fiber. Now this junction between them, that space is called the synaptic cleft. And this plate on the muscle fiber is called the motor end plate. Is everybody with me so far? Okay. So we've seen how the action potential comes down, triggers the calcium channels to open up, Calcium rushes in, triggers the exocytosis of the acetylcholine. Acetylcholine gets exocytosed into the synaptic end, or the synaptic cleft. Now, once in the synaptic cleft, all the acetylcholine are these red dots right here. So all these red dots, the acetylcholine is in a hurry. It's going to go and find the motor end plate. On the motor end plate, you have the sarcolemma, right? Everybody recall what the sarcolemma is? So it's going to find the sarcolemma. And on the sarcolemma, you have little gates. Can everyone see these green little gates? They're closed shut right now. So they're sodium ion gates, and they're closed shut. They're tightly closed shut. This acetylcholine is going to go to these sodium ion gates, and it's going to go bind to these sodium ion gates because they have little acetylcholine receptors. Once they go bind to these sodium ion gates, these sodium ion gates open up. What do you think is going to happen when these sodium ion gates open up? All this sodium that's hanging out in the synaptic cleft is going to rush into the muscle fiber. All of it is going to rush into the muscle fiber. So let's recap from the top, because repetition is key. Action potential reaches the synaptic end bulb, triggers calcium gates to open up. Calcium rushes into the synaptic end bulb, triggers the exocytosis of acetylcholine. Acetylcholine enters the synaptic cleft, binds to the acetylcholine receptors onto the sodium ion gates of the motor end plate. Sodium ion gates open up, and sodium rushes into the muscle fiber. Now, something's got to get rid of that acetylcholine. It can't just hang out there in the synaptic end bulb, right? So enter an enzyme called acetylcholine esterase. Anything that ends with an ACE is an enzyme. Enter enzyme called acetylcholine esterase that is going to eat up all the used acetylcholine. It's going to get rid of all of this used acetylcholine. So here's a nice zoomed in version of the synaptic cleft. So acetylcholine is going to bind to the sodium ion gates. The green dots are the sodium ions. Now you can see from the synaptic cleft how all the sodium ions are opening up and going inside of the muscle fibers right here. Now this can also happen with the botulism toxin. The botulism is a Botox. Botox is a subcutaneous injection that blocks the release of acetylcholine. So that is how it causes relaxation of muscles and therefore can cause a temporary anti-wrinkle properties in it. Now the story continues. What happens? So now we have seen this electrical signal jump the synaptic cleft and go from the neuron to the muscle fiber. Now the story is going to continue. The sodium has now entered the muscle fiber. Where is it going to go to from here? From here, it is going to do something. So the release of acetylcholine. We've talked about what happens right here. We've talked about the release of acetylcholine. This is all just repetition right here. Repeat, repeat, repeat until you get bored, and repeat some more. So we've talked about nerve signal opens up, calcium flows down the concentration gradient, acetylcholine into the synaptic cleft, crosses the sarcolemma into the muscle fiber, influx of sodium into the muscle fiber, acetylcholine esterase degrades acetylcholine. So is everyone with me so far? All right. So now the sodium has entered the muscle fiber. So now the sodium quickly spreads over the sarcolemma. Everybody still remembers what the sarcolemma is. Enters the T-tubules. Does everybody remember what the T-tubules are? Now it enters the T-tubules and goes over the sarcoplasmic reticulum. What is stored in the sarcoplasmic reticulum? Calcium. It goes over to the calcium reservoir and releases calcium. It opens the T-tubules, which are the gates. Once the T-tubules are opened up, the gates of the calcium reservoirs are opened up. So now, inside the muscle fibers, all of the calcium channels are opened up. So follow along the pointer here. All the calcium channels opened up, and there's a flooding of calcium inside the muscle fibers. Now, once there's a flooding of calcium inside of the muscle fibers, what do you think is going to happen? Remember your thin filaments and the regulatory proteins troponin and tropomyosin and how they had calcium binding sites? Exactly. That is what is going to happen. So calcium is going to go and bind to the troponin. Now, once calcium goes and binds to the troponin, the tropomyosin is going to change its shape. Once tropomyosin changes its shape, it is going to expose the myosin binding sites on the actin. Once the myosin binding sites on the actin are exposed, what do you think is going to happen? The golf club shaped heads of the myosin are free to bind to these little brown dots on the actin. This is what is going to happen. They are free to bind. Now, once they bind, something called a power stroke is going to take place. So here's what happens. In the contraction cycle, 4 steps are involved. And this process is an active process, meaning it requires a pretty heavy amount of ATP. So ATP is hydrolyzed, and it orients the myosin head. So the golf club shaped heads has to orient against the actin molecule for binding. Now, once the myosin head binds, a cross bridge is formed, and a power stroke takes place. So it binds, and a power stroke takes place that pulls the actin head towards the M line. So imagine the golf club shaped head pulling the thin filament towards the M line. So it's a tug of war. It's pulling the thin filament towards the M line. The next step is detachment of actin from myosin. Again, the ATP binds, it's an active process. And the golf club shaped head has to detach from the actin, and the process begins again. And then you have another power stroke, and that happens until the muscle is ready to relax. So let's look at the ATP hydrolysis. So an ATP molecule is used up. You have these 2 golf club shaped heads. They orient this way. ATP is hydrolyzed to form ADP. And these 2 golf club shaped heads have binded to the binding sites, and a cross bridge has formed. So a cross bridge has formed right here. In the next step, there is a power stroke. So you can see right here, this is going to move. So the M line, imagine the M line right here. So this is going to move towards the M line. So this is why the sarcomere shortens. So it slides towards the M line. It slides towards the M line. In the next step, it detaches, and it has to detach so that it can reattach to the following binding site and form a cross bridge again. So the cycle continues. It's a cyclic cycle. So this illustration explains the entire cycle. So myosin heads hydrolyze ATP, orient, power stroke happens, and so cross bridges take place, power stroke happens, detachment takes place, and the whole process continues until the muscle is ready to relax. Now, relaxation involves one major step. So it involves cutting off the calcium. So once the calcium is cut off, once the calcium is removed from the troponin, it involves the tropomyosin changing conformation again to block off the myosin binding sites. Once that happens, the nerve stimulation ceases to happen. The acetylcholine esterase breaks down any acetylcholine, the sodium channels close up, and muscle relaxation takes place. The muscle fiber returns to its resting length, and contraction of the antagonistic muscles takes place. The next concept that we're going to go over is the length-tension relationship. So what is tension? The amount of tension depends on what the resting length of the sarcomere is. So optimum resting length is what produces the greatest force when the muscle contracts. And the amount of tension generated depends on the amount of overlap between the thick and the thin filaments. So look at this graph right here. On the x-axis is the resting sarcomere length, and the y-axis is the tension developed. So the optimum tension is when the sliding between the sarcomere is between 2.2 micrometers. So when there's an overstretched sarcomere, you don't have optimum tension because there's weak contraction and few cross bridges can form. So over here you have the length of the sarcomere is about 3.8 micrometers, and you can't form, the myosin and actin cannot form optimum cross bridges because the sarcomere has overstretched. When the sarcomere is understretched, over here the sarcomere is only 1.8 micrometers long, there cannot be cross bridges optimally because they're too close to the Z discs, and they can't slide too much because they're already too close to the M line as well. So for optimum contraction, they have to be about 2.2 millimeters in length. The next concept is rigor mortis. So rigor mortis is stiffening of the human body about 3 to 4 hours after death. And that happens because of deteriorating sarcoplasmic reticulum. So what happens to muscles after a person dies? Now, after a person dies, they do not generate ATP. So the sarcoplasmic reticulum releases calcium, and this causes the muscles to contract. However, due to lack of ATP, the calcium cannot go back into the sarcoplasmic reticulum and therefore cannot contract, because muscle contraction is an active process as well. And hence, muscles contract after a person's death but cannot relax. And that is rigor mortis. Fibers and muscle fibers remain contracted until the myofilaments decay, which is about 24 hours unless temperatures are below freezing. The next concept we're going to go over are a few definitions. The first definition is a muscle twitch. A muscle twitch is contraction of one single muscle fiber, and it is the smallest possible muscle contraction of one single motor unit. It's not perceptible. Now, one single muscle contraction takes about 50 milliseconds. So in this graph, X-axis is time in milliseconds. Y-axis is force of the contraction. So latent period is the time that it takes for between the dance of the acetylcholine esterase, dance of the neurotransmitters, to release of the calcium from the sarcoplasmic reticulum. So it takes about 10 milliseconds from when the action potential reaches the synaptic end bulb to the dance of the neurotransmitters, to when sodium influxes into the muscle fiber, to release of the calcium ions from the sarcoplasmic reticulum. The contraction period is when the power stroke is taking place and the tension develops. Calcium binds to the troponin, the tropomyosin is shifting, so that is when tension is developing. And relaxation period is when calcium is going back into the sarcoplasmic reticulum and tension, we are losing tension and going back towards normal muscle tone. So that whole period of a twitch takes place in about 50 milliseconds. Next concept is isometric and isotonic contractions. An isotonic contraction is when the length of a muscle changes when it is contracted. If there is shortening of a muscle, it is called an isotonic concentric contraction. So when you flex your elbow, your bicep muscle is undergoing isotonic concentric contraction. When you extend your elbow, your bicep is undergoing lengthening, so it is undergoing isotonic eccentric contraction. An isometric muscle contraction is when there is no net change in length. So this is when you're holding a heavy textbook in place. So it is when your muscles are just not changing in length. It's for joint stabilization.